everyone. Before I begin with this topic of this session, I have a small scenario for you all to imagine. Now just think of this. You and your friends are supposed to go to Goa, and none of you have any clue about how you're supposed to travel there, where you're going to stay, and what are all the different places you're going to visit. So now there are two possibilities. One possibility is you could probably or your friend could take the lead to find all the relevant information required for the trip and then form an itinerary, which is possibly going to take one day or maybe some more days. The other possibility is you could probably take the help of a travel agent or a travel planner. Now he or she might already have an entire itinerary ready, so all you have to do is just pack your bags and just travel along with your friends to Goa. So this procedure happens in a very short time. Today's topic, which is general enzymology, I'll be talking about enzymes. Now enzymes is more or less like a travel agent or a travel planner. An enzyme is known to cut short the duration of several reactions that occur in our body. Now what are these reactions? These reactions are known as biochemical reactions or metabolic reactions which occur in our body. Let's have a look at what do you mean by enzymes as such. So enzymes is defined as biocatalyst. Now if you split the term, you have two parts. One is bio and the other is catalyst. Bio means life, which means that enzymes, they are synthesized and they exist within living tissues. The other part, which is catalyst is a substance which increases the rate or the velocity of a reaction. Now since enzymes occur in living tissues, the reactions which occur in living beings are known as biochemical reactions. So enzymes increase the rate or the velocity of biochemical reaction without itself undergoing any change during the process. So this is the definition of enzymes. Now you have to know that enzymes are pretty much just proteins. They are made of several amino acids which are linked together by a peptide bond. So since enzymes are proteins, they share the same physical and chemical properties as that of proteins. So they are heat sensitive, they are water soluble, they can be easily precipitated by certain protein precipitating agents such as trichloroacetic acid, sulfosalicylic acid. And one more point you have to remember is that at least 16% of their weight is made of nitrogen. The same properties are shared by enzymes and proteins. Now coming to classification of enzymes, there is an organization called as International Union of Biochemistry and Molecular Biology. It's also known as IUBMB, which is concerned with naming and classifying enzymes. Now as a general rule, the IUBMB says that the name of the enzyme should often end with the suffix ASE. Okay, so you have lipases, you have ureases, all these name of enzymes end with ASE. However, there are certain exceptions where the name doesn't end with ASE, such as trypsin, pepsin, chymotrypsin, etc. These are all enzymes, but you see that their names don't end in ASE. Another point to remember is that most often the enzyme names will often resemble the substrate on which it acts or the reaction which it catalyzes. So in case it resembles the substrate on which it acts, for example, I'll give you a simple example of glucokinase. This is an enzyme which bears the substrate, the name of the substrate on which it acts. Glucokinase is known to act on the substrate glucose. So this is one example based on which how enzymes are named. The other possibility, as I have already mentioned, is that it could resemble the reaction which it catalyzes. For example, lactate dehydrogenase. So you can see in the name itself, dehydrogenase, that is removal of a hydrogen group. That means the enzyme lactate dehydrogenase is involved in catalyzing oxidation reactions. So this is how enzymes are named. Other than naming enzymes, the enzymes are also allotted certain unique identification number, where the unique identification number starts with the alphabets EC followed by 4 digits. EC stands for Enzyme Commission. Now what are these 4 digits? So the first digit usually represents a class of the enzymes. Second digit denotes the subclass. Third digit denotes the subgroup. And the fourth digit denotes the number of the particular enzyme in the list. Or in other words, how the enzyme appears in the list. So Now I'll just give you a simple example of an enzyme called as hexokinase. Hexokinase is known to catalyze the conversion of glucose to glucose 6-phosphate, which is this. And during the process, ATP is utilized and it is converted to ADP. So you can see in this reaction, phosphate from ATP is donated to glucose, so that glucose forms glucose 6-phosphate. This is a phosphorylation reaction. Now hexokinase has the unique identification number of EC2711. Now let's see how did this number come into being. So starting with the first digit, which represents the class of the enzymes, as such, according to the IUBMB classification, broadly enzymes are classified into 7 major classes. So hexokinase comes under class 2 enzymes. Class 2 enzymes are known as transferases. So as the name suggests, transferase, these enzymes are involved in the transfer of a particular group to the substrate so that a product is formed. What is being transferred? In this case, you can see that phosphate from ATP is transferred to glucose so that you get glucose 6-phosphate. So that forms the basis for the second digit, which is 7, that is the 7th subclass under transferase enzyme. So here in case of the subclass under transferases, you have different subclasses which represents what groups are being transferred. So the first subclass under transferase belongs to the methyl group which is transferred. Likewise, the 7th subclass under transferase represents phosphate group being transferred. So that's how the 7 number comes here. The next one is 1, the first subgroup. How did this come into being? Now you know phosphate is being transferred from ATP to glucose. So the subgroup usually denotes what is the group present in the substrate which accepts the transferred group. Meaning glucose, has 6 carbon atom. To the last carbon atom, you have C2OH. Okay, an ethyl alcohol group over here, alcohol group you can see. So what happens is that phosphate goes and gets attached to the alcohol group. So this is the transferred group. And the alcohol group, which is no longer there now, is the acceptor group which is present in glucose. So we'll call this as an acceptor group. So the first subgroup usually denotes that there is an alcohol group which has accepted transferred group. So it basically depends on what group in the substrate has accepted the transfer group. And the last digit, that is 1, denotes the order in which the enzyme has been listed under the subgroup. So hexokinase is the first enzyme which appears in the list. So this is how the number or the unique identification number of an enzyme is given. Now we'll move on to the next thing, that is enzyme classes. Now all these years the IUBMB had come up with 6 major classes. Only very recently, about probably 2 years ago, the 7th class was introduced. So as such, we have 7 major classes now. They are oxidoreductases, you have transferases, hydrolases, ligases, isomerases, ligases, and finally the new one, that is translocases. Now you have to remember the enzyme classes in this particular order. You cannot jumble up the whole order. There's an easy way to remember this order. I have a mnemonic for you all, which is over the hill, life is lovely and terrific. Okay. This basically sums up the entire classes, and you can remember this mnemonic or you can come up with your own. You're free to do that. So this is how you're supposed to remember the major 7 classes of enzymes. Now let's have a look at each of these class of enzymes individually along with examples under each. So starting with oxidoreductases. Oxidoreductases are class 1 enzymes. As the name suggests, these enzymes are involved in oxidative reductive reaction. So it causes oxidation of one substrate with simultaneous reduction of another substrate or a coenzyme. Now, what do you mean by oxidation and what do you mean by reduction? Oxidation is nothing but a process in which oxygen is added or hydrogen is removed or electrons are removed. Reduction, on the other hand, is a process by which hydrogen is added or electrons are added or oxygen is removed. So now let's have a look at a simple general formula which explains the basis on how oxidoreductases act. So imagine you have a substrate which is AH. You can see that hydrogen is attached to A. That means this substrate is said to be in reduced form. Okay, and then you have another substrate which is B. Hydrogen is not there, it's not attached, so this is said to be in oxidized form. Now, in the presence of class 1 enzymes, that is oxidoreductases, what happens? You will see both happening, oxidation and reduction. So, AH, which was in the reduced form, gets oxidized to A. Hydrogen is removed. This becomes the oxidized form. And simultaneously, B, which was in the oxidized form earlier, gets reduced to BH. So what you can see here is both processes happening simultaneously. You have oxidation reaction occurring here and you have a reduction reaction happening here. So this is what oxidoreductases do. Now let's have a look at the two examples under oxidoreductases. I have mentioned lactate dehydrogenase and succinate dehydrogenase. We'll have a look at lactate dehydrogenase first. So lactate dehydrogenase acts on the substrate lactate and it converts it to pyruvate. So initially lactate is said to be in the reduced form. It loses hydrogen to form the oxidized pyruvate. Okay, so what is, at the same time you can see that there is a coenzyme which accepts the hydrogen from lactate, which is NAD+. NAD+ accepts the hydrogen and gets converted to NADH. So you can see NAD+, which was earlier in the oxidized form, it gets converted to NADH, which is the reduced form. Hydrogen is added to NAD+. So you can see simultaneously both oxidative and reductive reactions occurring in this process, and the enzyme which is responsible for the conversion of lactate to pyruvate is known as lactate dehydrogenase. Now we'll have a look at the second one. This is an enzyme which acts in one of the steps of citric acid cycle. So succinate dehydrogenase acts on the substrate succinate and converts it to fumarate. The hydrogen from succinate is accepted by a coenzyme known as FAD, that is flavin adenine dinucleotide. Hydrogen gets attached to FAD to form FADH2. And the enzyme is known as succinate dehydrogenase. So what you can see here, you can see succinate being converted to fumarate. This is an oxidative reaction, and simultaneously you can see FAD getting converted to FADH2. This is a reductive reaction. So this is it about oxidoreductases. You have other examples also under this class of enzymes that I have not mentioned here, such as oxidases, peroxidases, and also reductases. The next class of enzymes is transferases. As the name suggests, these enzymes are involved in transfer of a particular group to the substrate. Now the group that is transferred could be anything. It could be a methyl group, it could be a carbon dioxide group, a amino group, or a phosphate group. So you have a lot of possibilities that can happen. So a general formula that can explain this whole process is this. Imagine you have a substrate A and then you have BX. okay, and X is the molecule that is supposed to be transferred. So in the presence of class 2 enzymes, that is transferases, the group which is X gets transferred to A plus B. So you can see how this group has been transferred from one substrate to another. This is the basis for the action of transferases. You have examples under that. The first example that I mentioned here is kinase. So there are several kinases that participate in various biochemical reactions in our body. One of the most common example is that of hexokinase. Hexokinase catalyzes the first step in the reaction glycolysis, where you can see that glucose gets converted to glucose 6-phosphate. This occurs in the presence of the enzymes hexokinase. Kinases as such, all kinase enzymes are known to transfer a phosphate group. Remember that. So the phosphate group is usually donated by ATP, adenosine triphosphate. Once the phosphate group is donated, it gets attached to glucose to give you glucose 6-phosphate, which means that phosphate is attached to the 6 carbon atom of glucose, and simultaneously ATP gets converted to ADP. So this is an example of a transferase reaction. Likewise, you also have another example which is called as transaminase or aminotransferase. As the name suggests, this enzyme is involved in the transfer of an amino group, which is NH2. Now, the most common or the most important aminotransferase, you have something known as AST and ALT, alanine transaminase and aspartate transaminase. I'll just talk about alanine transaminase. In the reaction, what happens is you have alanine, which is an amino acid. It combines with alpha ketoglutarate, which is a keto acid, in the presence of the enzyme aminotransferase, also known as alanine aminotransferase or alanine transaminase, represented as ALT. So during this process, alanine gets converted to pyruvate and alpha ketoglutarate gets converted to glutamate. Pyruvate is an alpha keto acid again, and glutamate is an amino acid. And this reaction occurs in the presence of a coenzyme known as PLP, also known as pyridoxal phosphate. So this is an example in which you can see an amino group which is present in the amino acid alanine. It gets transferred to alpha ketoglutarate, which is a keto acid. Thereby, because of the transfer of the amino group, alpha ketoglutarate gets converted to glutamate to become an amino acid, and simultaneously you see alanine, which is an amino acid, it gets converted to alpha keto acid due to the removal of the amino group. So this is an example of a transferase enzyme again. You have several other enzymes which I have not listed under the set of examples here. You have transmethylases, which is involved in the transfer of a methyl group. You have transcarboxylases, which is involved in the transfer of a carboxyl group. So this is it about transferases. Now I'll move on to the next class of enzymes, which are hydrolases. As the name suggests, hydro meaning water. So water is used to cleave a substrate so that you get products. Okay, and usually an ester, ether, peptide or a glycosidic linkage or a bond is broken down with the help of water. Now you have different examples under hydrolases. Most common are the digestive enzymes. Digestive enzymes such as enzymes that are concerned with protein, carbohydrate or lipid digestion, all of them are examples of hydrolases. Now I'll give you a simple example of that of sucrase enzyme. Sucrase enzyme is a digestive enzyme and it acts on the substrate sucrose, which is a disaccharide. A disaccharide containing glucose and fructose. It is a non-reducing disaccharide. So in the presence of sucrase, water molecule is utilized to cleave the disaccharide so that you get individual products which are nothing but glucose and fructose. So this is one example of a hydrolase. Another example, as I mentioned here, is urease. So urease acts on urea. A water molecule is required. It cleaves urea, which is a waste product from protein digestion. Urea is broken down into ammonia and carbon dioxide. So again, this is an example of a hydrolase where water molecule is required to split the product. So this is it about class 3 enzymes. We'll move on to the next one, that is class 4 enzymes. Now, in lyase, what happens is it's almost similar like a hydrolase, but the only difference is water is not used here to break the bond. Instead, some other mechanism is used to break the bond or to form any double bonds between the products. So, examples that you can see here is aldolase and argininosuccinase. Aldolase is an enzyme which participates in glycolysis. So, you have fructose 1,6-bisphosphate. This is a 6 carbon containing sugar. So fructose 1,6-bisphosphate is the substrate here. It is acted upon by aldolase. Aldolase cleaves fructose 1,6-bisphosphate to give you two products so that the carbon atoms are equally distributed between the two products. So you get glyceraldehyde 3-phosphate. This is one of the products formed. This is a 3 carbon containing sugar, and simultaneously you also have one more, that is dihydroxy acetone phosphate, which again is a 3 carbon containing sugar. So this is an example of a lyase. Another example, as you can see, is argininosuccinase. This is an enzyme in the citric acid cycle. Again, there what you can see is you have the substrate argininosuccinate. Acted upon by argininosuccinase. Argininosuccinate is then cleaved to give you two products. One is arginine and the other one is fumarate. So this is again another example of a lyase. Now we'll move on to the next one, that is class 5 enzyme, which are nothing but isomerases. Now before I move on to the examples and the definition, you need to know what you mean by isomers. Isomers are any compounds which have the same molecular formula but different structural formula. So these enzymes are involved in the interconversion of isomers. Now, the examples that I mentioned here are phosphohexose isomerase and triose phosphate isomerase. Both these enzymes take part in glycolysis. So I'll just show you the first example, that is of phosphohexose isomerase. This enzyme acts on the substrate glucose 6-phosphate. Okay, glucose 6-phosphate is converted to the product which is fructose 6-phosphate. In the presence of the enzyme phosphohexose isomerase. Now, how are these isomers? Now, glucose 6-phosphate and fructose 6-phosphate. You can see there are two sugars over here, glucose and fructose. They are both hexoses, that is containing 6 carbon atoms, and they have the same molecular formula, that is C6H12O6. Now, they differ, they become isomers with respect to their functional group. Now, as such, the functional group which is present in glucose is an aldehyde group, whereas in fructose, the functional group is a keto group. Hence, glucose is known as an aldohexose and fructose is known as a ketohexose. So they are both functional isomers. And this is how isomerase enzyme acts on the substrate glucose 6-phosphate and converts it to its corresponding isomer form. The next one is triose phosphate isomerase. As the name suggests, it acts on a 3 carbon containing sugar. So again, this enzyme participates in glycolysis. You have the substrate glyceraldehyde 3-phosphate, which is a 3 carbon containing compound. It is acted upon by triose phosphate isomerase. I'll just write TPI over here. So glyceraldehyde 3-phosphate is converted to dihydroxyacetone phosphate, which again is a 3-carbon containing compound. Again, you can see that these are two isomers. They have the same molecular formula. However, they differ with respect to their functional group. Glyceraldehyde is an aldotriose, whereas dihydroxyacetone phosphate is a ketotriose. So they differ with respect to their functional group again. So apart from isomerases, you have other examples like epimerases, racemases, mutases. All these examples come under isomerases. Moving on to the next class of enzymes, which are ligases. Ligase, the term ligate, which means to join. So these enzymes are involved in catalyzing reaction, which involves in joining of two molecules with the help of energy in the form of ATP. So I'll just give you two examples, that is acetyl-CoA carboxylase. This is an important step in fatty acid synthesis. So what happens in that case is you have acetyl-CoA, which is a 2-carbon containing compound. It combines with carbon dioxide. Now these are the two molecules that are supposed to be joined. You have 2-carbon containing acetyl-CoA, which combines with 1-carbon containing carbon dioxide. The enzyme is called as acetyl-CoA carboxylase. And as the definition says, you need energy, so ATP is utilized for the joining of the two molecules. So you get eventually something known as malonyl-CoA, which is a combination of both acetyl-CoA and carbon dioxide to give you a 3-carbon containing compound. And then malonyl-CoA is utilized for fatty acid synthesis. So this is an example of how two molecules are joined with the help of energy. Another example is in glutamine synthetase. So glutamine synthetase participates in certain protein metabolism reactions. So in this, what happens is you have glutamate, which is an amino acid. It combines with ammonia, and then you have the enzyme glutamine synthetase. I'll just write GS. Energy is required in the form of ATP. These two combine together to give you glutamine. So this is it about class 6 enzymes. We'll move on to the next one, the last one, which is translocases, the new class of enzymes recently introduced. So these enzymes, they catalyze the movement of ions. It could be any type, or molecules, could be carbohydrates, proteins, or lipids, across the membranes or their separation within the membrane. So either it could move from the cytoplasm to the mitochondria, or vice versa along the mitochondrial membrane. That is one example how a translocase could act. So one example is that of proton pumps. Proton pumps are involved in pumping out hydrogen ions. Again, that is an example of a translocase. And then you have carnitine acyl carnitine translocase. This helps in the transport of acyl carnitine. This is very much important, especially in utilization of fatty acids for energy purpose during beta oxidation. So during the process, you can see carnitine and acyl carnitine, which is a fatty acid attached to carnitine. They get transported across the membrane, mitochondrial membrane, so that energy is later on produced through beta oxidation. Types and structure of enzymes. Now, as I've said earlier, enzymes are mainly proteins. They're made of several amino acids linked together by a peptide bond. So the different types of enzymes, there are two types mainly, simple and complex. Simple enzymes are nothing but the protein part of the enzymes. Okay, and complex is the protein part. You have the protein of the enzyme. In addition to that, you will have a non-protein part which is also attached, which is known as cofactor. So that brings us to the other term, that is holoenzyme, which you will need to know. Holoenzyme is known as the functional unit of enzyme. Holoenzyme is nothing but the complex enzyme as such. Protein part, which is known as the apoenzyme. and a non-protein part which is known as a cofactor. So the cofactor could be loosely or tightly bound to the apoenzyme, and it is mainly required for the activity of the enzyme as such. Now let's have a look at what are the different types of cofactors which are found or known. So cofactors as such I've already mentioned, they are the non-protein molecules required for enzyme activity. They are divided into two major categories. They could be either coenzymes or metal ions or activators. Now coenzymes are cofactors which are organic in nature. You have different types like NAD+, FAD+, TPP. I'll come to this later in the next few slides. And then metal ion activators which are inorganic in nature. You have magnesium ions, manganese, zinc, etc. So let's have a look at coenzymes first. Coenzymes are non-protein organic molecules required for the activity of the enzyme. They attach to the apoenzyme. You have two groups of coenzymes. You have group 1 and group 2. Now group 1 are those coenzymes which are involved in reactions that allow the transfer of hydrogen or electrons. That is nothing but oxidative reductive reactions. And group 2 you have coenzymes which are involved in reactions that involve the transfer of another group other than hydrogen or electrons. Now let's have a look at what are the different examples of group 1 and group 2 coenzymes. So coming to group 1, which is mainly involved in transfer of hydrogen or electrons. So if you have a look at all the coenzymes, you will notice that most of the coenzymes are derived from vitamin B. So we'll start off with the first two coenzymes. You can see nicotinamide adenine dinucleotide, which is also called as NAD+, and nicotinamide adenine dinucleotide phosphate, also represented as NADP+. These two coenzymes are involved in oxidative reductive reactions, and they are derived from the vitamin niacin, which is also known as vitamin B3. The next two set of coenzymes under group 1, you have flavin mononucleotide and flavin adenine dinucleotide, also represented as FMN or FAD. These two are derived from riboflavin, which is a vitamin B, also known as vitamin B2. So these are examples of group 1 coenzymes. We'll move on to the next group, that is group 2. So as you can see here, some other group is being transferred and it is not hydrogen or electrons. Again, if you go to see the first example of coenzyme, which is thymine pyrophosphate. Thymine pyrophosphate is usually seen in the transfer of a hydroxyethyl group, that is C2OH. Again, this coenzyme is derived from the vitamin thiamine, also represented as B1. The next coenzyme, which is pyridoxal phosphate, it is involved in the transfer of an amino group. You would have seen it earlier in the previous slides when I was talking about aminotransferases. So aminotransferases uses the coenzyme PLP or pyridoxal phosphate, and the group which is transferred is an amino group. Again, this is derived from the vitamin pyridoxine, which is vitamin B6. Then you have biotin. Biotin is involved in carboxylation reactions. So it is involved in the transfer of carbon dioxide, and the enzyme which catalyzes these reactions are called as carboxylases. And again, this coenzyme is derived from the vitamin biotin, also represented as B7. The next one is coenzyme A. It is involved in the transfer of acyl groups of fatty acids as such, and it is derived from pantothenic acid, which is vitamin B5. Then you have tetrahydrofolate, which is derived from folic acid, again another vitamin, which is B9, vitamin B9. Tetrahydrofolate is involved in the transfer of 1 carbon groups. And then last but not the least, you have adenosine triphosphate, also called as ATP. It's the energy currency of the cell. It is responsible for the transfer of a phosphate group Usually kinases utilize ATP as a coenzyme, and this is not derived from any vitamin. So with this, we are done with the classification of coenzymes. We'll move on to the next, that is activators. Activators are non-protein inorganic molecule, again attached to the apoenzyme and is required for the enzyme activity. So you have certain enzymes which require metal ions for their activity, and these ions are tightly bound to the apoenzyme. Those enzymes are known as metalloenzymes. Now let's have a look at what are the different examples of activators. So you can see zinc over here. Zinc is required for the enzyme carbonic anhydrase. This is responsible for acid-base balance. Then alcohol dehydrogenase responsible for detoxification of alcohol. Then DNA polymerase required for DNA synthesis. Then magnesium is required for these two enzymes, hexokinase and phosphofructokinase. Both the enzymes are involved in glycolytic reactions. Manganese again is required for the first step in glycolysis, that is catalyzed by hexokinase. Then you can see copper is required for tyrosinase. This is mainly for melanin production. And then iron, which is required for catalase and peroxidase. Both the enzymes are required for detoxification of hydrogen peroxide, which is a free radical. And then you need molybdenum, which is required for xanthan oxidase. Xanthan oxidase is required for uric acid formation in the body, which is one of the products formed during the breakdown of purines. And then you have chloride, which is required for amylase. Amylase is an enzyme which is required for the breakdown of starch or glycogen in the body. So these are the common activators involved in various biochemical reactions in our body. Now we'll move on to prosthetic group. Now whatever I've spoken about coenzymes and cofactors, all these cofactors are loosely bound to the apoenzyme. However, there are certain cofactors that may be tightly bound to the apoenzyme through the process of formation of covalent bonds. Now, prosthetic group as such cannot be easily separated from the apoenzyme, and if there is any absence of the prosthetic group, or if the prosthetic group is separated from the apoenzyme becomes inactive. So common example is that of heme, which is a prosthetic group. You might have heard of hemoglobin, which is made of heme and globin. So heme is the prosthetic group which is present there. Now there are certain enzymes that may contain heme as a prosthetic group. Heme is mainly comprised of iron and a porphyrin ring structure. So this is how heme is composed of. Heme is seen in these enzymes, that is cytochrome oxidase, that is involved in electron transport chain, and then again you may also see it in catalase and peroxidase. Both the enzymes are involved in detoxification of hydrogen peroxide, which is a free radical. So with this we are done with cofactors. We'll move on to the next one, that is mechanism of enzyme action. Now it is very important to know how enzymes act, how it converts a substrate to the product. There are various theories which explain this mechanism, but some of the most important theories I have listed over here. We have Michaelis Menten theory, lowering of activation energy, Fisher's template theory, and Koshlins induced fit theory. Now let's have a look at each of these theories one by one, starting with Michaelis Menten theory. Now before I begin with each of those theories, I would like to explain a little bit about the enzyme structure and what do you mean by substrate. Now the enzyme usually, as I've said earlier, is a protein, and it consists of a site, a cleft or a crevice, that is this, which is known as the active site. Now the active site may be comprised of two parts. So the two parts are known as substrate binding site and you have a catalytic site. These are the two parts of the active site. Now what do you mean by a substrate binding site and what do you mean by a catalytic site? Now substrate binding site is a site of the active site where the substrate goes and binds. What is a substrate? Now this is a substrate which binds to the active site. A substrate is a substance that has to be converted to product eventually. So this is what do you mean by substrate binding site? It's the site at which the substrate binds. The next part is the catalytic site. Catalytic site is mainly involved in catalysis, that is conversion of substrate to product. So these are the two divisions of the active site. Now it is not necessary that both the substrate binding site and the catalytic site be at the same place. What can happen in certain enzymes is you may have the substrate binding site at one position of the enzyme and you may have the catalytic site towards the other side of the same enzyme. So the sites may vary or it can be at the same site. So this is about active site and substrate. We'll move on to the first theory, that is Michaelis Menten theory. According to Michaelis Menten, it is said that when an enzyme binds with a substrate S, it forms an enzyme substrate complex. Okay, so this enzyme substrate complex is quite unstable. So eventually, over a period of time, enzyme substrate complex decomposes to give you two things. One is a free enzyme and then the substrate which is there gets converted to product. So this is Michaelis Menten theory. Enzyme combines with substrate, gives you enzyme substrate complex, dissociates because it is highly unstable to give you a free enzyme and then product. Moving on to the next theory, that is lowering of activation energy. Now, whenever a substrate has to be converted to product, two things have to occur. First thing is the substrate or the reacting molecules have to be in close proximity with each other so that bond formation can occur or bond breaking can occur so that product is formed. So remember, the proximity is very important. Apart from that, the substrate molecules or the reacting molecules should attain some minimum energy so that it can react and form product. What is that minimum energy? That minimum energy is known as a activation energy. So, it is said that whenever a substrate forms a product, it forms an intermediate called as a transition state. So the transition state is a highly unstable state again, and eventually it dissociates to form a product. Now, if you plot a graph between the free energy and time required for the reaction to occur, this is in case of an uncatalyzed reaction where the enzyme is not participating in the reaction at all. So initially, when the substrate enters into the reaction, it is said to be at a very low energy. Say for example over here, this is S or substrate. The substrate has a very low energy and it is said to be in ground state. Eventually what happens is, say for example, the temperature of the biological system is increased. So the substrate tends to get some kind of energy from that increase in temperature, attain some kind of kinetic energy, and so the free energy tends to increase till it reaches a maximum point. The point at which the maximum energy is attained is called as a transition state. Okay, so once this transition state is attained, next thing what happens is the transition state dissociates and then forms a product. So what is seen later on is the energy of the transition state slowly tends to decrease till it reaches a point which is lower than the energy attained by the substrate. This is the point at which the product is formed. So I'll just draw a line from the ground state. You can see that the energy of the product, free energy of the product, is lesser than the free energy of the substrate. Now this is in case of a uncatalyzed reaction, and if you try to subtract the free energy of the transition state from the free energy of the substrate, that will give you the activation energy. So activation energy is the difference between the free energy of the transition state and free energy of the substrate. and it is the minimum energy required for the substrate to be converted to product. Now this whatever I have spoken was in an uncatalyzed reaction. Now let's see what happens when an enzyme comes into play. So when an enzyme comes into play, you will see that the free energy of the substrate increases, reaches a maximum point over here so that a transition state is attained, and then you can see there is a decrease in the free energy to finally give you product. Now if you minus this transition state, the energy of the transition state and the energy of the substrate, you will get an activation energy. Again, this is for an enzyme catalyzed reaction. You will notice that the activation energy of an enzyme catalyzed reaction is much more smaller in comparison with the activation energy of an uncatalyzed reaction. So as a result of which the whole reaction progresses at a much faster pace. Now you can also try to see this as a mountain. Like say for example the uncatalyzed reaction, you can just imagine this to be a big mountain. Okay. Now if you take a car and you start from point S to point P, it will take you a long time to ascend the mountain, to climb up the mountain and then come down. But if there was a tunnel through the mountain, it would have been much easier to cross from point S to point P. So in the presence of an enzyme, a tunnel is formed through the reaction so that the entire process is cut short and it occurs at a much faster rate. So this is the whole mechanism of lowering of activation energy in the presence of an enzyme. Now we'll move on to the next theory, that is Fisher's template theory. According to the theory, the active site of the enzyme is very rigid. So I'll just draw the enzyme along with the active site. So whenever a substrate has to bind, it has to bind to the active site of the enzyme. Now as per the theory, the active site is quite rigid. So only a particular substrate who is complementary to the active site of the enzyme can bind to the enzyme. So if you have a substrate which is identical to the active site of the enzyme, then only the substrate goes and binds to the active site to form enzyme substrate complex. This is the substrate. Yeah, you have the enzyme substrate complex, which will eventually disintegrate or dissociate to give you the free enzyme. And then you have the product which is formed. However, if there was a substrate which was not identical to the active site, say for example, if this was the scenario, you had a substrate which was not complementary to the active site, the shape of the active site of the enzyme, the reaction wouldn't have taken place. So very often Fisher's template theory is also called as a lock and key model. Why? Because you know that only a particular key that fits into the keyhole of a particular lock can open the lock. Likewise, only a particular substrate which is identical to the or complementary to the active site of the enzyme can only bind to the enzyme and then form product. So which is why it is also called as a lock and key model. Now, Fisher's template theory is not acceptable because it doesn't really explain a lot of the properties of enzyme catalyzed reaction. So, we have a better theory which explains all the properties of enzyme catalyzed reaction, and it is called as Koshlins induced fit model or fit theory. So, as per this, the active site of the enzyme is highly flexible. It can easily transform itself so that it fits the substrate. So the substrate doesn't have to be identical to the active site of the enzyme. I'll just draw a substrate over here which is not at all identical to the active site. Okay. In the presence of the substrate, the active site tends to undergo certain conformational changes so that it tries to accommodate the substrate. So eventually you get an enzyme substrate complex. And you can see how the active site has changed from a triangular cleft into a rectangular cleft so that it accommodates the substrate. It forms the enzyme substrate complex. Eventually this disintegrates or dissociates to give you the free enzyme, where the active site is retained, goes back to its original form, and then you have the product which is formed. So this was it about Koshlins induced fit model. Now Koshlins induced fit model explains a lot of the properties of enzymes, such as enzyme inhibition, how enzymes are inhibited and how they are regulated. Everything is explained by this particular theory, which is why Koshlins induced fit model is also known as a hand and glove model, meaning you might have seen this examination gloves in the OPD, in the outpatient department, where one single gloves can fit so many different shapes of hands and so many different length of fingers. So likewise, the active site can undergo different changes or undergo configurational changes so that it can easily fit the substrate according to the needs. So that was it about Koshlins induced fit model. So those were the theories that explains the mechanism of action. Now we'll have a look at what are the different factors which affect enzyme activity. So the rate or the velocity of the enzyme catalyzed reaction is affected by four main factors. As you can see here, enzyme concentration, temperature, pH, and finally substrate concentration. We'll have a look at enzyme concentration first. So as per this, it is said that the rate of the enzyme catalyzed reaction increases proportionally with increase in the enzyme concentration, provided the substrate concentration is not limiting. There should be sufficient amount of substrate present so that the reaction can occur. Imagine you have these many enzymes present in the system and you have only limited substrate present. So there's no point, you will not have much of the reaction occurring. The reaction will stop as soon as the substrate is utilized, which is why it is very important that sufficient substrate concentration is present so that the reaction can occur. And this property is mainly used to estimate various serum enzymes that is involved in diagnosis of disease. You might have heard of enzymes like alanine transaminase and aspartate transaminase, which is used to check out how well the liver is functioning. So likewise, there are different serum enzymes that can be utilized for diagnosis of various diseases that occur in our body. So I'll just draw a graph between enzyme velocity and the enzyme concentration. You will get a linear graph which indicates that with increase in enzyme concentration, you will get a proportional increase in enzyme velocity. This rule will be followed provided you have enough amount of substrate present. Next, moving on to the next factor, which is temperature. So according to this, the rate or the velocity of an enzymatic reaction will increase with increase in temperature till it reaches a specific point, that is a maximum point called as an optimum temperature. Furthermore, if you try to increase the temperature, there will be a decrease in the velocity of the reaction. So that is how the velocity of the reaction and the temperature are interrelated. Now there is a terminology called as temperature coefficient, also known as Q10. What do you mean by that? So with every increase in 10 degrees Celsius of temperature, you will notice that there will be an increase in the velocity of reaction at least 2 times. It gets doubled with every 10 degree increase in temperature. Now I told you there is an optimum temperature by which your velocity of the reaction reaches a maximum. So that optimum temperature of most of the body enzymes is around our normal body temperature, that is around 37 degrees to 40 degrees Celsius. However, there are certain exceptions, that is in case of Taq polymerase. This enzyme is used in polymerase chain reaction. The optimum temperature in case of that enzyme is around 100 degrees Celsius. It's quite high. Now we'll have a look at the graph if you plot it between the velocity of the reaction and temperature. Like I've said, the optimum temperature is somewhere around 37 to 40 degrees Celsius. So what you'll notice first is with increase in temperature, you will find that the velocity of the reaction increases and it reaches till a point, a maximum point, which will correspond to the optimum temperature. So this is the optimum temperature. Beyond the optimum temperature will result in decrease in the velocity of the reaction, giving rise to a bell-shaped curve. Now, why do you see such a pattern? What happens is, initially when the reaction occurs and when the temperature is increased, the reacting molecules or the substrate attain certain kind of energy from the increase in temperature, known as kinetic energy. So because of increase in kinetic energy, more and more substrates tend to collide with each other and eventually ends up in formation of products. So this is what happens when you increase the temperature till it reaches a maximum velocity at the optimum temperature. Any further increase in the temperature beyond the optimum temperature will result in loss of the enzyme activity, resulting to decreased formation of products. Now, why there is a loss in the enzyme activity? I have mentioned at the beginning of this session that enzymes are proteins and they follow the same physical and chemical characteristics of a protein. So just like proteins, where the protein structure can be lost, especially when it's subjected to extremes of temperature or extremes of pH, resulting in a process called as denaturation, where the activity is lost. Likewise, enzymes also will undergo certain denaturation processes, eventually leading to loss of its structure and activity. Thereby, the substrate cannot bind to the active site of the enzyme and product is not formed. So which is why you can see there's a decrease in the rate or the velocity of the enzyme catalyzed reaction beyond the optimum temperature. So this was it about the effect of temperature. We'll move on to the next one, that is effect of pH. Now the pattern seen here is almost similar to the pattern seen in effect of temperature. You will notice that there is an increase in the rate of the enzymatic reaction with increase in pH till it reaches a maximum point at the optimum pH. Further increase in pH beyond the optimum pH will result in decrease in the rate of the enzymatic reaction. Now, again, the optimum pH of most of the enzymes lies in the range of 6 to 8. However, you have certain exceptions like that of pepsin, which is an enzyme that is required for protein digestion. The pH that is required is towards the acidic side, that is around 1 to 2. Then there is alkaline phosphatase. Again, this requires a pH of 9 to 10, an alkaline pH. And acidic phosphatase again requires an acidic pH that is around 4 to 5. But otherwise, generally, most of the enzymes act best at 6 to 8 pH. So we'll just have a look at the graph when you plot it between enzyme velocity and pH. I'll just write down the pH over here. 6, 8, 10 and 12. And as you know, the optimum pH is somewhere between 6 to 8. So what happens is when the pH increases, you will see that the enzyme velocity also increases till it reaches a maximum point which corresponds to the optimum pH, somewhere between 6 to 8. Furthermore, with increase in pH beyond the optimum pH, there will be a decrease in the enzyme velocity so that you get a bell-shaped curve. Again, here you can see that there is a decline in the velocity of the reaction beyond the optimum pH. That is because, again, enzymes being proteins, they undergo denaturation with extremes of pH. Too acidic and too alkaline pH also can affect the enzyme activity. So that is why the activity is lost. The active site will not be available so that substrate can go and bind to it and product is formed. Hence, you will find a decrease in the velocity of the reaction after it crosses the optimum pH. So that was it about the effect of pH. Moving on to the next one, that is effect of substrate concentration. So as such, for a given enzymatic concentration, the rate of the enzymatic reaction increases initially with increase in concentration of substrate. Then towards the end of the reaction, what is noticed that even with further increase in substrate concentration, there is no further increase in the velocity of the reaction. So initially when you see, the reaction curve is almost linear and then later on the reaction curve flattens out. So I'll just show you the graph. This is a graph between enzyme velocity and substrate concentration. When the reaction begins, you will notice that with increase in substrate concentration, the velocity of the enzymatic reaction also increases. So this increase is proportional, hence you will get a straight graph, a linear graph. Afterwards, what is noticed that with increase in substrate concentration, you will get an increase in enzyme velocity, but the increase is not proportional. Okay, so this is the second part of the graph. Increase in enzyme velocity is not proportional to the substrate concentration. Then towards the end, you will notice that any more increase in substrate concentration will not result in any increase in the velocity of the reaction. You will see a flat graph after that. So if you have a look at the entire graph, you will notice that it is a rectangular hyperbola. And if you try to study the graph also and look into the enzyme kinetics, you will realize that this first half of the graph, where there is a proportional increase in enzyme velocity along with substrate concentration, the reaction kinetics is said to follow first order kinetics. In the second half of the graph, where you will find an increase in enzyme velocity, but the increase is not proportional to the substrate concentration. So in that case, this part of the reaction is said to follow mixed order kinetics. And eventually, towards the later half of the reaction, the later end, you will notice that no matter how much you increase the substrate concentration, there is no further increase in the enzyme velocity. So, enzyme velocity is independent of the substrate concentration, as a result of which zero order kinetics is followed here. So, this is the graph for effect of substrate concentration. So based on the previous graph, Michaelis Menten has come up with an equation so that it links the velocity of the reaction with the substrate concentration. So according to the equation, it says that V, or velocity of the enzymatic reaction, is equal to V max, which is the maximal velocity attained by the entire reaction, and then into substrate concentration divided by Km. Km is nothing but Michaelis constant, I'll come to this later, plus substrate concentration. This is the equation given by Michaelis Menten. Now, coming to Michaelis Menten constant, let us consider at a given point the velocity of the reaction V is supposed to be at half maximal velocity, half V max. And you know that the equation, Michaelis Menten equation, is V is equal to V max into substrate concentration divided by Km, Michaelis constant, plus substrate concentration. Now, if you substitute the value of V into equation 2, you will get half V max, this is the value of velocity of V, is equal to V max into substrate concentration divided by Km plus substrate concentration. Now you can cancel V max on either ends. You will get half is equal to substrate concentration divided by Km plus substrate concentration. So if you cross multiply, you will get Km plus S is equal to 2S. Bring the substrate towards the other side, you will get Km is equal to 2S minus S, which is nothing but S only. So the main thing that you have to remember at the end of it is, at half the maximal velocity, the Michaelis constant is equal to the substrate concentration. So this brings us to the definition of Michaelis constant, which is nothing but the substrate concentration at half the maximal velocity. It is often expressed as moles per liter, and the main features of this constant is, you will notice that it is a characteristic feature of an enzyme. just like how we have fingerprints. Human beings have fingerprints which help distinguish between one person to another. If you notice, even twins will not have identical fingerprints. Even twins will have completely different set of fingerprints. Likewise, each enzyme will have a particular Km value. Also, it varies from enzyme to enzyme, and it can also vary from enzyme. If supposing an enzyme acts on multiple substrate, the Km value for each of the substrate will be different for that particular enzyme. And then it also measures the affinity of an enzyme for its substrate. It is said that the Km value has an inverse relationship with the affinity of an enzyme to a substrate. So if the Km value is low, the affinity of an enzyme to the substrate will be high. If the Km value is high, then the affinity of an enzyme to the substrate is low. And last, it also determines the amount of substrate concentration required for significant catalysis. How much should be the concentration of substrate so that the substrate can be converted to product. So that is it about Michaelis constant, and usually the values range between 10 raised to minus 5 to 10 raised to minus 2 moles per liter. So with this, we are done with this session. I would like to summarize this session and tell you what exactly I have covered. So I started off with the definition of enzymes. I've told you that they are biocatalysts. There are 7 major classes of enzymes based on the classification given by IUPMB. So you have oxidoreductases, transferases, hydrolases, liases, isomerases, ligase, and the last one, which is a new one, that is translocases. You have 2 types of enzymes, simple and complex enzyme. Simple enzyme is just the protein, whereas complex enzyme, you have the protein part. Additionally, you have a non-protein part. So that brings us to hollow enzyme, which is nothing but a complex enzyme made of apoprotein, the protein part, and a cofactor, which is the non-protein part. Cofactor again is divided into coenzymes and activators. Then I spoke about the mechanism of action of enzymes. There are 4 types. We have Michaelis Menten theory, lowering of activation energy, Fisher's template theory, Koshlins induced fit model. Moving on to the factors affecting enzymes activity, there are 4. You have effect of enzyme concentration, effect of temperature, effect of pH, and effect of substrate concentration. Based on the last property, that is effect of substrate concentration, I spoke about the Michaelis Menten equation, which links the velocity of an enzyme catalyzed reaction with the substrate concentration. So you have the equation V is equal to V max into substrate concentration divided by Km, which is the Michaelis constant, plus substrate concentration. So coming to the last thing, that is constant Km or Michaelis constant, it is nothing but the substrate concentration at half the maximal velocity. So with that, we are done with this session.